Thank you for choosing to watch this video, which will describe the steps required to perform the enema tip adherence testing required for the HPTN 106 study. This video is provided as an instructional aid only. Procedural instructions provided in the laboratory section of the SSP must be followed. Throughout this procedure, there is a high risk of contamination from one sample to the next. Sterile techniques must be adopted to reduce the possibility of contamination. The use of a biological safety cabinet, however, is not required. Participants will be provided with a 50 ml conical tube that has been pre-labeled with the unique participant identification number. They will also receive a prepaid and addressed padded mailing envelope in which to return the used enema tips. Participants are instructed to place used enema tips into the pre-labeled conical tube and to add the date that the tip was used onto the tube. The tube should be placed into the prepaid mailing envelope and returned to the clinic so that it arrives within 3 days of use. The used enema tips should arrive at the CRS lab in the prepaid mailing envelope. Inside the envelope will be a conical tube containing the used enema tip. The conical tube should be labeled with the unique participant identifier and the date of use. Document this information along with the date and time that the enema tip was received on the appropriate study log as indicated here. This document can also be used as a testing plate map or Collie plate map. It has 8 rows, A through H, and 12 columns and has been designed so that it matches the testing plate layout. Use this Collie plate map to document the required information for all used enema tips received in the lab. Up to 5 enema tips can be tested together on a single Collie plate. This Collie plate map will be used later in the process as a guide when pipetting samples, and for reading and recording test results. Returned enema tips should be stored at ambient temperature and must be tested within 7 days of use. Up to 5 enema tips can be tested in each batch, but tips should be tested as soon as possible after they are received. Each batch of testing will include 3 negative controls and between 1 and 5 used enema tips. Negative controls are used in this process to detect any possible contamination in the work area. Note: there are no positive controls used in this procedure. Start by labeling one 15 ml conical tube for each control or sample to be tested. Control tubes should be labeled exactly as written on the plate map. Unused swab, bench swab, and unused enema tip. Sample tubes should be labeled with the unique participant ID and the date of tip use. Using sterile techniques, including clean gloves, pipette 1.6 ml of sterile water into each of the labeled 15 ml conical tubes. The use of filter tips is essential for this process. The first negative control is an unused swab. Remove a new swab from its packaging and place into the appropriately labeled conical tube containing the sterile water. Carefully cut the shaft of the swab so that the shaft will fit inside the tube. Replace the cap on the conical tube. The next negative control is for the bench swab. Using an unused swab, wipe around the surface of the bench or absorbent material around the area where testing will take place. Place the swab into the appropriately labeled tube containing sterile water, cut the shaft and replace the cap onto the conical tube. The final negative control is an unused enema tip. Remove an unused tip from storage for this purpose, remove and discard the green cap. Using an unused swab, wipe the swab around the enema tip a minimum of 5 times. Place the swab into the appropriately labeled 15 ml conical tube containing sterile water, cut the shaft and replace the cap onto the conical tube. Prepare all 3 negative controls before moving on to any participant samples. If, during any stage of this process, gloves or equipment has become contaminated, change gloves and decontaminate the equipment following local guidelines. Before opening the participant sample, double-check the participant information written on the tube against the collie plate map to ensure that the details match and that the correct sample has been selected for testing. Also compare information on the participant sample with that written on the conical tube that contains the sterile water. Carefully remove the participant sample from the conical tube. Remove the green cap from the enema tip and discard. Examine the tip and determine, in your opinion, if the tip appears to be visually soiled. Document this as a yes or no on the collie plate map. Swab the used enema tip thoroughly a minimum of 5 times, making sure to include all external surfaces including the neck or wide area of the tip. Discard the used enema tip. Place the swab into the appropriately labeled conical tube containing the sterile water. Cut the shaft and replace the cap onto the conical tube. Repeat the process for any other used enema tips that need to be tested. To minimize contamination, gloves should be changed, and scissors should be decontaminated after each participant's sample has been handled. After all control and participant tubes have been processed, vortex each tube for a minimum of 20 seconds. Tubes should then be placed in an incubator set to 37 degrees Celsius. Incubate the tubes for between 60 and 90 minutes. After this time, remove the tubes from the incubator. It is good practice to document the time when all of these testing steps took place, as well as the name of the person who performed these steps. Space is provided on page 2 of the collie plate map for the documentation of these items. After incubation, we are ready to start preparing our collie plate. As mentioned earlier, the collie plate map is a visual guide that will help us. Here the negative control, unused swab will be placed into row A, wells 1 through 6. The negative control, bench swab will be placed into row C, wells 1 through 6, and so on. You will see that wells 1 through 6 of rows B and D are left unused. This is to minimize contamination. Starting with the first negative control labeled unused swab. Remove the cap from the conical tube and discard the swab. Pipette 250 microliters of the control solution into wells 1 through 6 of row A. The same pipette tip can be used for each of the 6 wells. The use of filter tips is essential at this stage to help minimize contamination. Note that the collie plate lid is being used to cover up some of the wells and other rows or columns to help minimize contamination. Move on to the next control in the sequence and pipette according to the plate map using a new pipette tip when moving to the next specimen. It is recommended that the pipette be decontaminated and that gloves are changed between participants' samples to further reduce the risk of contamination. After all samples have been pipetted into the collie plate, replace the collie plate lid and place the plate into the incubator set at 37 degrees Celsius. Incubate the plate for between 12 and 36 hours. This incubation can occur overnight. When the incubation is complete, remove the collie plate from the incubator. Place the plate onto a white surface in a well-lit area for reading. The good lighting and white background are essential for an accurate reading of the plate. Orient the plate so that it matches the plate map. A negative test result is indicated by the absence of a blue color in all 6 wells. As you see here, solution corresponding to the unused swab negative control wells, 1 through 6, of row A, remains colorless and should be documented as negative. The other two negative controls have also given negative results and should be documented as negative on the plate map. For our participant sample in row F, all 6 wells have turned blue, so this will be reported as positive. Document the results for the participant sample as positive on the collie plate map as indicated. Even if only one well is blue, the result should be reported as positive. In this example, we see that only 2 of the wells in row D have turned blue. This should be interpreted as a positive result. As you can see here, placing the plate on a clear white background makes the wells easier to read. As previously mentioned, page 2 of the work map is used for documenting details about the assay performed. The timing of procedures performed, and who performed those procedures should be documented. It is recommended that the final reading and result interpretation step be double-checked or verified by a second person, although this may not always be possible. The form also has a space to document any comments that are relevant to the procedure. Completed plate maps are source documents for this adherence testing and should be securely stored according to your site procedures. Important. If any of the negative control wells have any blue color, this is a sign of contamination, and the run must be interpreted as invalid. Continue to document the results of all controls and participant samples onto the plate map, but immediately notify the HPTN Lab Center.